Our next speaker is a scientist at Karolinska Institutet, Sara Hägg. She works with how to measure aging reliably at the cellular level. Now, this is crucial if we're ever going to develop drugs targeting aging and extending the healthy lifespan. So, Sara, please go ahead.
Yes, thank you, Linus, and the team for inviting me. And not so easy to go up after Peter's very nice presentation. I'm more of this boring scientist person, I would say. So anyway, I'm going to talk a little bit about some of the research and my perspective to aging, and some of the research we do in my group. But I'm also going to end with lining out some problems or really where we can use AI and also giving examples of where we're currently using these type of methods in the research related to things that I do, because this was also specifically asked for in this forum. All right, but I think we need to start from the beginning again. What really happens in humans as we age? And of course, you all heard about this already, and I don't need to do the full introduction. We've had many speakers mention this, because the first things that happens in our bodies are really the molecular and cellular changes. And they are best illustrated by the different hallmarks of aging that have been defined now for more than 10 years, and they are expanding. But my point here is that this is the current view that we're using as an illustration. It's not the full view, obviously. There must be many, many more processes that we are not currently aware of and that we haven't really mapped yet. All right, but this is really the molecular and cellular things that change with aging. This is something that starts already in utero, and then we can follow that across the whole lifespan. But of course, when does it transition from going from growth and development into something that we call aging? That is something that we don't really have well defined today. Because of course, we don't talk about children as being individuals that age. We talk about growth then in that case. But it's actually the same type of processes that is ongoing in the body across the lifespan. But of course, when we get a bit older, we can also start to map aging in other ways in the body. And then we see things where we start to have symptoms. So changes that started as cellular and molecular processes that then manifest on a tissue level in specific organs in specific parts of the body. And we can also talk about functional aging. We can measure function in many different ways in the body. So as we know, for example, at mid-age, we need glasses, our eyes start to change. Even earlier than that, we can see aerobic capacity going down in the body, and etc. So there are a lot of functional things that change with age, and we can start to measure them, but later in life. So the molecular and cellular changes, we can map them across the whole lifespan. But many of the functional parts, we have to map them as they start to deteriorate in our bodies. And of course, eventually, we will end up with a more multifactorial part of aging, where we see systemic aging across the whole body. We get multimorbid, we develop diseases and become frail. So this is really more towards the end stage. But actually, all of this is aging. All of this is human aging, and we can map this in many different ways. So a lot of what we do in my research is also trying to think about how can we actually measure this in humans and follow individuals over time to really understand more about the aging process. And as you understand, because there are so many different ways, aging is a complex phenotype, and we can map this in so many ways. There are also many, many ways that we can measure this. And as I told you, the cellular processes you can follow across the whole lifespan, whereas other things, cognitive decline, there is no use in starting to map this already early in life because there is no variation, really. the big variation starts when you starting to see decline in a population. So that will be something that you map later in life. Whereas aerobic capacity, you can start to map that already in the 30, when people are around 30. Now there is an initiative in the academic world called the Biomarkers of Aging Consortium. They actually have a meeting, their second conference next week at Harvard, where I'm going. But they do a lot of work because what we don't have in this field are guidelines. We don't have standardized measures like we have in many other medical fields where clinical biomarkers are well known and mapped and used in the clinic. Okay, I think it's starting to, let's see how it works. So for aging, we need to do this work as well. It may not be so fascinating and the most fun part, but this is really what we need to do, and trying to do it also within the whole community of researchers so that we can get everyone involved to standardize. What do we need to do to measure biomarkers? How can we do it in a systematic way across different cohorts, across different settings, across different parts of the world? So this work needs to be done because ideally what we want is to use these type of measurements and biomarkers in many studies where we can do clinical research as well, so that we can really have evidence-based medicine where we can also test different interventions, treatments, lifestyle choices, etc., in real clinical studies. But then we need to know how to measure effects. Because as we heard also from earlier speakers, we can't really use the hard endpoint mortality. We need to use surrogate markers like these biomarkers so we can connect that to also aging in humans. And of course, because we can measure it in so many different ways, we also need to standardize and think about how we do this. And we know that people that then have a higher biological or accelerated aging phase, they will also have increased risk for developing a lot of age-related diseases and earlier mortality in life. But the way that we assess these changes into a biological age marker could be done in many different ways. So here is one example where we already see machine learning methods and AI tools being helpful in terms of developing these type of measurements. And what we do then is that in some way we have a composite score of different markers, different assessments, and we put them together, and in that way, within a population, we can say something about the deviation. We call it the age gap, the age deviation, the age delta, where people can have either a more positive profile, so having a lower biological age, or a higher accelerated profile of biological age. So this is one example where we do already see the help of machine learning tools. And here is another example where they've also mapped, we already heard today, and I will come back to that, a lot of these different omics techniques that we can use. So here we have examples, epigenomics, transcriptomics, proteomics, metabolomics, etc. So these are different levels of biomarkers that you can measure in blood or in other tissues in humans. And from all of these different assessments, you can create different aging clocks using different types of methods, like machine learning. And it has been done in many different ways from many different groups. And as you can see, they are in different ways related to mortality or obesity, body mass index, and different diseases. So we know that in general, these different types of aging clocks generated from different types of data, they work to assess health in humans. So what we do in my research is that this is an example from a longitudinal study called SATSA that we have collected at the Karolinska Institutet, where we actually measure a lot of different markers. So we have everything from cellular and molecular markers to functional markers, clinical markers, and also markers at the end point when we talk about functional aging and frailty. So we do this assessment and we map it longitudinally in this cohort. And this is from a paper some years back now, but here you can really see what it looks like within a cohort when you try to map all of these things simultaneously. So we call these spaghetti plots because one spaghetti line illustrates the trajectory in one individual. So if you have multiple measurements, you can see that the trajectories, the spaghetti is going up and down. So this is, of course, why we need standardized tools. How should we do this in the best ways? Because a big problem in the field is also the measurement techniques. There's a lot of errors, technical variability in the measurements that we need to handle. So I would say that a lot of the variability you see here in trends going up and down could be biology, but it's also a lot of technical noise that we want to get rid of. But here you can see then on the top row, and all the DNA methylation assessments are done from epigenetic clocks. And then we have physiological age, which is a clinical biomarker-based clock. And on the further down, it's all the functional markers of aging. So, of course, functional markers like cognitive decline is something that goes down with age, then, obviously. But you can see that it's quite a big variability within these as well. So what we then have done is trying to see as well, okay, but if we now can map these biological ages longitudinally in the cohort, what happens if we can also assess other things in the cohort? How does it influence the trajectories, these different lines, spaghettis, in different individuals? So one thing where we've done a couple of studies is where we try to understand if a person started to take a treatment for something, a person with hypertension will get a treatment for that. What happens then to these trajectories, these lines of biological aging? And can we learn something from that? It's not a clinical trial, but we can learn from following people repeatedly over time. So for example, if you can then have different trajectories here and really find out what influences these different changes. Is there a specific treatment that is more beneficial to another? So for example, if we do have these spaghettis and then we know this person started this treatment here and we can see a change to the trajectory, it would indicate that there is a beneficial effect on the biological aging from that intervention. And one example here is where we looked at, in this longitudinal SATSA cohort, we looked at physiological age. So this is predicted biological age using clinical biomarkers. And we only looked at people that at some point across these end of the life course in the old ages, from 50 years to 90 years, they were all at some point statin users. So statin is a treatment for high blood cholesterol levels. Okay, so here they are. You can see the spaghettis going up and down. But what happens if we try to center on the time when they started the treatment with statin use? So this is what we see, basically, as illustrated in the cartoon before, that you dramatically change your trajectory when you start a specific treatment. So it probably means that you somehow have beneficial effects beyond just the treatment indication. And this is, I usually say that when we speak about biological age, what I mean is that it's something that relates to health. What is the health of different individuals? And it's a different way of looking at this. So meaning that we can see this similar trends with other treatments where you actually show that, for example, being a pre-diabetic person that is not treated is worse compared to a diabetic patient that has had the treatment, because you normalize, you standardize values. And this is what we can also see on a lot of different measurements on biological aging. And then, of course, we wanted to do it a little bit more sophisticated. So I'm not going to explain the model, but this is a more advanced statistical model where we can also adjust for a lot of things. But ideally, what this means is that we can compare within the persons what happens in this person before and after a treatment. And we try to adjust and take care of everything else that could contribute to this changes on the biological age. And what we then saw was that for certain different types of drug treatments, we can actually see a beneficial effect across a lot of spectrum of different measurements of biological age. So here in green for calcium channel blockers, for example, which is a treatment for hypertension, we can see a general beneficial effect both on functional aging, on frailty, and a lot of different epigenetic clocks that is generated from epigenetic data. So across both cellular, molecular, and functional assessments of aging, there is a total beneficial effect then when you start to treat your high blood pressure in the body. And specifically for calcium channel blockers, we're now doing some follow-up work with collaborators because it's also understood that some of the off-target effects, so beneficial effects beyond the indication of calcium channel blockers, may be that it's actually working to treat senescent cells. So we already heard about senescent cells before. So more work to be done here. How am I on time? Where is my... Okay, I'll wrap it up. So this is, I'll just end with highlighting some AI methods that could be useful in this field. So for example, here's another ongoing work in my study where we looked at many of these, again, assessments of biological age from different domains. And we can use machine learning methods to try to combine these different biomarkers to identify people that belongs to a high-risk group or a low-risk group. And this is what we've done using data from the UK Biobank, which is a very big study from the UK. And we then followed them over time. And just using markers of biological age like this, we can identify who is more likely to die sooner than later because of these differences. Another example of AI methods is, of course, prediction models using image data. So this is an example from a colleague at Copenhagen University. So here they used deep neural networks to predict morphology from human fibroblasts. And when you do this, you can actually predict how senescent these cells are from these stained images. And then that is directly related then to a biological age predictor of senescent cells. And they map that to different diseases as well. So this is one area where AI tools is very popular in medicine for image predictions. And my final example I would end with is just this integration of data layers. Lots of people have talked about it already. I just want to illustrate that this is a general problem for many things, but also for aging, where we follow people over time because we want to map the longitudinal changes. So we have repeated assessments. It can be different animal models. It can be humans. And then, of course, we have all of these layers of the omics data that we talked about. So this already is a very complex problem, and we may just be taking samples from blood here. But then the next steps, what is actually happening now in the field is that we're trying to create biological age clocks for different organs so that it's not just a universal clock, it's very different for different parts of the body. So you can do this in different organs and imagine all the different data layers you can then create from different organs. And adding to this is also then the single cell resolution. There is a lot of work ongoing on molecular research where we want to map things that happens in each single cell from different tissues. So imagine again, different organs and then the different cells in these organs and the different omics techniques for each of these different alternatives. So in the end, of course, we end up with so much data that is so complex where we try to map the aging over time. And this is really where we need the help of AI methods to really try to get into the complexity of all of these data. So with that, I thank you so much for the time.